Good morning.
Good
Good
morning,
morning,
sir.
Good morning,
sir.
sir.
Okay, so today we have 19 students on board. Fine. So in the last lecture, we have completed with the quaternary structure of proteins. So as I said, we'll be starting with the presentation. So today we'll have a presentation by whom? I think it's Sanskruti.
Yes, sir.
Yeah. So Sanskruti will be talking on.
Shall I start with
Yes.
protein folding, and factor affecting
Tell
on
me.
protein folding?
Yes. Yes. Okay. So you can
Shall I
start with your.
start?
Yes, yes. You can present your
Okay.
screen and start the presentation.
Is it visible?
Yes, it's
Yes, it is.
Okay. Uh, Good morning. Today I will be telling about protein folding and factor affecting and on protein folding and protein stability. In the first slide, protein folding. Protein folding is a process by which polypeptide chain are folded and become a biologically active protein. Proteins are held together by various molecular interactions. Those interactions having two types, that is covalent and non-covalent. Non-covalent is weakly bonded, and covalent are strong bonded interactions. Then during translation, each protein is synthesized as a linear chain of amino acid or a random coil, which does not having a stable 3D structure, and they don't show any specific function. The amino acid in the chain eventually interact with each other to form a well-defined folded protein. Protein of folding of a protein into their correct native structure is a key function to their is a key to their function. Failure to the folding is cause number of diseases. Uh, in this picture, we can see how protein are folded into 3D structure. Uh, this is a linear This is a linear chain of amino acids which get coiled into 3D structure. When a protein get coiled from when protein get coiled from linear to a 3D structure, then follow some stages which are primary, secondary, tertiary, and quaternary. Now, four stages of protein folding. Folding of protein is a complex process which involves four stages that give rise to the various 3D 3D protein structure essential for a diverse function in human body. The structure of protein is hierarchical arrangement from primary to quaternary structure. A wide variation in amino acid sequence account for different conformation of protein structure. In this slide, we can see primary structure. Primary structure is nothing but linear chain of amino acids which are joined together by peptide bonds. This is the uh, first stage of protein folding. Then secondary structure is the next stage. Secondary structure is generated from formation of hydrogen bond between atom in peptide backbone, which fold the chain into their alpha helix or beta sheet. In the picture, this part which shown by green color is alpha helix, and which is in blue, this is beta sheet, which are present in secondary structures. Then is tertiary structure. Tertiary structure is formed by folding of the secondary structure sheets or helix into one another. The tertiary structure of protein is geometric shape of protein. It is usually has a polypeptide chain as a backbone. Which one or more secondary structure, the tertiary structure is determined by interaction and bond of amino acid side chain. Tertiary structure hydrophilic interaction that sulfide and hydrogen. Next, if we see quaternary structure, quaternary structure results from folded amino acid sequence in tertiary structure in which
So
each
slowly your
other
voice
to
is
give
breaking.
rise to a functional protein. Yes, sir.
Your voice is breaking.
Oh, sir, I tried to change my position.
Yeah.
Now is it fine, sir?
Yes, yes.
Okay. Then quaternary structure. Quaternary structure results from folded amino acid side chain in tertiary structure. Interact further with each other, give rise to a functional protein. Well-known example of quaternary structure is nothing but hemoglobin or DNA polymerase. Protein get fold in four different stages with the different interactions in between them. Some experiments have been done with protein folding. First is Assenson's experiment in 1950. Christian Ascension conduct a series of experiments in which he determined all information in which he determined all the information that needed to fold a protein in 3D form. In this experiment, he followed following steps. As first, he denatured the ribonuclease. By for denaturation, he used reducing agents, that is urea and mercaptoethanol, which due to this reducing agents, the polypeptide are not folded properly. They need a random coiling. Due to the random coiling, they do not show any activity. In next step, renaturation of ribonuclease is carried out by removing urea and mercaptoethanol. After renaturation, the refolded protein has natively folded, and this shows his function as a first. Here's a diagram. This is a native ribonuclease structure. This is a disulfide bond present in between ribonuclease. Reducing agents that is urea and mercaptoethanol, which reduce disulfide bond. Reduce disulfide bond due to reduction of disulfide bond. Polypeptide get random coil. Due to this random coil, they do not show any activity. Then again, renaturation by removing beta mercaptoethanol and urea. Then the protein is again folded into their native structure and shows her following functions. Then Levinthal paradox. Levinthal paradox states that the number of possible conformation available to a protein fold, for example, 100 amino acid sequence in protein. Then we have three different conformation. Another example is if we have a sample of chain 10 raised to 12 conformation, then it's take time, that is 5 into 10 raised to 35 second. That is that this time is required for his native conformation. It's a too large time. Then the conclusion is protein not a random. Protein folding is not a random, but they have a specific pathway. If they get random, then they require lots of times. So is it not possible? Then conclusion is folding not a random but have a specific pathway. Proteins are folded by using some models. Which models are as follow? First, framework model. Framework model. This suggests that folding is start with formation of secondary structure, which interact to form more than advanced folded intermediate. In picture we can see formation of individual secondary structure element. Next, molten globin, staining globin, and native and fully unfolded set of globular protein. Some characteristic of molten globule is the presence of native-like content of secondary structure, the absence of specific tertiary produced by a tight packaging of amino acid side chain, the absence of specific tertiary structure produced by a tight packaging of amino acid side chain, model for early stage of protein folding that is nothing but hydrophobic collapse. Next model is nucleation condensation model. Nucleation condensation is a sequence of nuclear in this sequence would have a native that which are present which are come to form nucleus, which formation of nucleus is hydrophobic residue. Protein folding, sometimes protein folding is associated with chaperones. Many molecular chaperones assist protein folding. Definition of chaperones is molecular chaperones are the proteins that assist the conformational folding or unfolding and assembly of other macromolecular structure. Chaperones is nothing but a proteins. Chaperones having two types. First, HSP70 or HSP40 or chaperones. Those HSP40 and HSP70 are translational proteins which are found in eukaryotics. Co-translation is that is they are come together or they are working to from translational process. Then next is chaperones. Chaperones is grow EL and HSP shows how grow is working. When uneaston folded protein is in contact with grow, then grow this step is reduced and get attached to upward of grow EL. This then this protein is get fold into grow EL. The grow EL temperature and pH, the protein get fold. Then ATP is hydrolysis, cap get open, and native folded protein is comes out. Here, this is the working of grow EL chaperone. The when get protein fold, many different factors are affect on protein folding. Protein folding that first is nothing. First is solvent. The presence of water molecule fulfills the availability hydrogen bonds that at those sites. Therefore, as a general solvent, water does not drive interaction of hydrogen amino acids, but also perform favorable enthalpic interaction between polar or charged group. Uh, then salt can act as a protein stabilizing or destabilizing agent depending on protein sequence and folded state topology. Salts affect on thermodynamics and kinetics of protein folding. Then next is temperature. Temperature protein generally unfold at temperature higher than basal temperature of organism. So the temperature is a very important part for protein folding. If temperature rise, then protein get unfold and denatured it. Next is presence of cofactors. During protein folding, many cofactors are present. The presence of cofactor often stabilize the native protein. They stabilize to the uh, native protein. The cofactor has ability to interact specifically with unfolded polypeptide. Third, the presence of cofactor sometimes essential for polypeptide to fold. Fourth, cofactor polypeptide can dramatically accelerate formation of functional protein. Then, most important part is molecular chaperones. They work as holders. They bind holding intermediate to prevent their aggregation. Well-known example of chaperone is DNA J, HSP33 or GROW-EL. The reason of the behavior is that protein folding is severely affected by heating, and therefore some chaperone act to prevent or correct damage caused by misfolding. Uh, Protein stability. Protein stability is a net balance of force which determine whether a protein will be its native folded conformation or a denature. Solubility can be influenced by a number of extrinsic or intrinsic factors. Many factors are affecting on protein stability, which are as follow. First, temperature. At the extreme temperature, protein get unstable. At extreme pH also affect on protein solubility, which can causes if pH get in extreme, then it's cause instability in protein. Organic solvent. The non-polar solvent utilized show the ability to enhance thermal stability of both proteins. Protection against the thermal denaturation was especially pronounced. Organic solvent is nothing but water and on solvents. Then cheotropic agents. Cheotropic agents are co-solutes that can disrupt the hydrogen bonding in between the water molecule and reduce stability of native state of proteins by weakening hydrophobic effect. A well-known example of cheotropic agent is nothing but urea and gonadium hydrochloride, which destroy the tertiary structure. Another part in protein stability is disulfide bond. If the disulfide bond are breaking, that is nothing but reduction of disulfide bond, and then the carboxymethylates with idoacetyl. The reduced protein is denatured, and they get unfolded. Due to this unfolding, ligands are not properly bind. Another point is ligand binding. Ligand binding increase the stability of a protein. Ligand able to bind with protein is nothing but ligands. Well-known ligand is O2 molecule that bind in hemoglobin or myoglobin structure. Uh, Here's the all references which I had used for make this presentation. Thanks for your patience. Any questions?
Okay, Sanskruti.
Yes, sir.
Yeah, a good presentation. So maybe initially she has given us an idea about the protein folding, the mechanisms which we have already seen in detail, but still she has given the glimpses. And at the end, she has touched upon the points that what are the factors which could be affecting the stability or the folding of this particular protein. Any questions from the audience? Yeah. See, I told you that this presentation should be interactive. You have to raise question. You have to go for discussion. If there are no questions, no discussion which is happening, I will stop the presentations then. Okay, Sanskruti, you are telling that you can use urea or gonadium hydrochloride or mercaptan ethanol. So what actually it does?
They are reducing agents, and they are unfold that ribonuclease, which are these ribonuclease get fold, and to unfolding this, they use reducing agent, which reducing agent is urea and beta mercaptoethanol.
Means what do they do actually? Means what kind of bonds
They
would
break. Huh. They break the disulfide bond, which present in between ribonucleases.
Okay, fine. See, when you're saying that a particular reducing agent is used, means you're having an environment where the hydrogen concentration is very high. Okay, so the disulfide bond, if it is present into a given protein, that disulfide bond could be broken in that reducing environment,
Yes, sir.
and this bond is broken and it is converted into SH. So that's the logic behind it. So if you have
Huh.
done some practical in page, or if you have gone for a DNA
SD.
isolation, you might have seen that you add mercaptoethanol into it. So this mercaptoethanol, it actually helps in denaturing the proteins. It breaks the hydrogen bonds, sorry, the disulfide bonds. And if the disulfide bonds are broken, means the other bonds, they are accessible to the environment, so that they can be easily broken, like hydrogen bond, hydrophobic bond, etc, etc. Right? So that's the reason behind it. In addition to that, we also add phenol and the various other chemicals. So now you can put on your logics into it. Fine. When you're talking about the factors which are going to affect your protein folding, so these factors could be dependent upon that interactions are happening within the protein when it is getting folded. So already we have seen the different type of the non-covalent interactions are there. Okay, so it could be your ionic bond, the hydrogen bond, hydrophobic bond, then the van der Waals bond, planar bond, etc, etc. So all those bonds, these bonds are the type which could be included into some electrostatic interactions, okay, or the bonds which are formed due to a change into the solvent of the environment, etc, etc. So those factors you can now talk about that they are going to affect the protein folding. So it could be your solvent. So as she has brought about some examples, that if you're using organic solvent, if you're using simply the water or the other type of the solvent, you can imagine that what effect it is going to happen onto your protein. So if you are using some highly polar type of the solvent, so obviously it is going to break all your electrostatic interaction. Okay, so the protein folding may not take properly there. If you are using only water, so in that case, although it will increase the hydrophilic interactions, but it will maximize the electrostatic interactions of that particular protein. Okay, so the environment or the solvent is very important here. Now the next two factors which are very important, that is the pH and temperature. So obviously, if you are going to change the pH of the solution, we have seen that this pH is going to affect on the amino acid residue, that is the specifically the R groups of the amino acid residues which are present into the protein, because the pH is going to change the nature of that R group. And if the nature is changed, nature in the sense, either it will be converted into the complex form, or it could be zwitterionic form, or it could be the anionic form. So it may get converted into any one of these form, and if there is a change in its nature, then obviously it is going to affect the protein folding. Okay, so pH is very important here. Then next could be your temperature. So obviously, if there is a change in temperature, it is going to break this non-covalent interaction, because these are the very weaker bonds. So even if there is a slight increase into the temperature, these bonds will be broken, and the protein stability cannot be achieved, or the protein folding will not take place. Now, one more thing which you can add into this, when you're talking about the temperature as a factor, that when you are increasing the temperature of the system, it is going to change the pH. Laksha diya. Okay, so a change in temperature is also affecting the pH of that particular solution. So it's a combination of this temperature and pH which is going to affect the molecule. So in this case, what we are talking about is the protein folding or the stability of that particular protein, right? So these two factors are very important, where you have to uh, emphasize onto this, where you should try to explain that what actually happens when you are changing the pH and the temperature. Fine. Then the other factors could be few of the factors what she has already told about. But one more thing which I can add here about is the entropy, the conformational entropy. See, what we want an extended conformation to be converted into a native conformation. Okay, so I told you when we were talking about the models of the protein folding. So the extended conformation is having a very high entropy. On the other hand, folded conformation is having a very low amount of entropy. So that free energy which is associated to convert an unfolded protein into the folded protein has to be taken into consideration. So that conformational entropy uh, is one more factor which we can talk about, which is going to affect your protein folding. And obviously the next point could be your the free energy which I told you, that is going to affect the protein folding and its stability, right? So, like this, with some good examples, you can explain this particular point if it is asked about the factors which are going to affect the protein folding or the stability. Yeah. Any questions where anyone want to ask something? Any difficulty in this point? What she has explained? Yes. Okay. So, if there are no further questions, a good presentation, Sanskruti. Still, you have a scope that you can improve upon. One more request to all of you. As I told you that you find a place where you can give a good presentation. You can have a practice whether your voice is properly uh, heard by others or there is a breaking, something like that. Okay, so just check that particular point. So the things there will be no problem when you're giving the presentation, right? So hardly it's a matter of 10 to 15 minutes where you're going to present. So kindly do that. Fine. Let's move to the next one. Who is next? I think it's Vaishnavi.
Yes, sir.
Yeah. So Vaishnavi, I think she's going to talk about the protein misfolding and the diseases which are associated with. Okay. You can present your screen and start the presentation.
Yes, sir. Is the screen visible?
Yeah.
Okay. Good morning, everyone. I'm going to talk about protein misfolding and the diseases. We all know that what is the protein folding, how the protein gets folded. So first, the polypeptide chain folds to become the biologically active protein in its native conformation, so that it function body. Then, then what is protein misfolding? That it can't achieve its native conformation or its 3D structure, and this misfolded protein gets aggregates, and these aggregations are called as amyloids, and the process is known as amyloidosis. We will see the amyloidosis in next slide. But first, what are the causes of misfolding and the aggregation? The first cause, obviously, mutation. We all know there is a mutation in gene sequence, then there is the a misfolding in protein. If there are second, if there are errors in process of transcription and or translation, there could be misfold of proteins. And then failure in folding of chaperones and of chaperone machinery or protein control mechanism. Then post-translational modifications. If there are mistakes in post-translational modification, there could be protein misfolding. So, what is amyloidosis? So it's if the protein gets aggregate, if the protein gets misfold and then it gets aggregates, it forms the um amyloid fibrils. So you can see in the image, this is normally folded protein. It gets misfold. When it misfold, it forms amyloid, and this forms stacks, and they are form amyloid fibrils. They are specially they have hydrophobic core, and hence they are insoluble, and there is a tissue, and they cause the tissue damage and functional compromise in the proteins and their function. If you can see in this picture, if the protein gets misfold, either it get for degradation, either it or it go for its phagocytosis or autophagy. You can see the proteosomes degrade the misfolded proteins in intracellular and extracellular. There is a autophagy where the autophagosomes degrade the misfolded protein or the aggregates. There are molecular chaperones which work on the misfolded protein. So the hydrophobic patches on the misfolded protein are exposed, and there is a ATP binding of ATP, and the hydrophobic patches are get inside the protein, and hence the protein gets folded properly. And there are proteasomes. They destroy the misfolded protein into small polypeptides. There is a ubiquitination method. Protein gets attached to ubiquitin gets attached to misfolded protein, and then it transferred to proteasome, and it gets degraded into short polypeptides. So this is what happens in normal circumstances. Intracellularly, proteasome pathway, and extracellular, there is a macrophages. And in amyloidosis, this control mechanism is failed. There are mutations, and therefore there is accumulation and aggregation, and they form the fibrils. What are the properties of amyloid protein? So physical, it's continuous, it's non-branching fibril, like you can see in this image. They are 7.5 to 10 nanometer in diameter, and there are cross beta pleated sheet conformation. Chemically, they are 95% fibrillar protein, 5% P protein, that is proteoglycan serum amyloid. Uh, in biochemical forms of amyloid, there are two forms: major form, minor form. There is AL protein, AA protein, beta amyloid protein, and in minor, there is transthyretin, beta 2 microglobulin, serum amyloid P. So, what AL protein causes? AL protein causes AL amyloidosis. It is caused by a bone marrow disorder. We all know that bone marrow produces cells in the blood system, including the plasma cells. And plasma cells are part of immune system, and it makes antibodies, immunoglobulins. Immunoglobulins composed of four chains, that is two light chains and two heavy chains. So what happens in amyloid patients? The plasma cells produce abnormal antibody protein, that is abnormal light chain protein, and the light chain becomes misfold and abnormal, and it results in the forming of amyloid fibrils. You can see in the image the bone marrow. There is a bone marrow produces plasma cells, but the light chain gets misfold. It gets abnormal, and it form amyloid fibrils, and these fibrils get deposited on various organs, heart, nerves, kidneys, liver. The symptoms are chest pain, fatigue, shortness of breath, low BP, and etc. Then AA amyloidosis. So, what is AA? So this, when there is a inflammation or infection in our body, our liver produces serum amyloid A protein. But if the infection or that inflammation lasts for the long period of time in our body, the this protein, serum amyloid A protein, is produced in large amount, and then the small portion of this protein, that is A protein, gets separated from serum amyloid A and deposits in tissue as AA amyloid. And this deposition is most in the all organs, but most in the kidney, and it causes nephrotic syndrome. And the symptoms are very similar to AL amyloidosis, like swelling of ankles and legs, low BP, enlarged spleen, liver, and thyroid. Then transthyretin. Transthyretin is a 127 amino acid protein made in liver. It transports the thyroid hormone thyroxine and vitamin A, that is retinol. Uh, Thyroid form fibrils in either because of genetic mutations either or either increased number of wild type thyroidin. Genetic mutation mutations, thyroidin amyloid, uh, it is it caused due to different
Just 1 minute.
mutations.
Ruchira, Vaishnavi, 1 minute. Ruchira, you have some problem? Ruchira, she
No,
was
sir.
raising hand.
No,
Okay,
sir.
okay, fine, fine. Vaishnavi, continue.
Yes, sir. There are in genetic mutations, different mutations lead to different symptoms, and it can pass from to the next generation. But if there is increased number of wild type thyroid, it's the normal thyroid protein becomes unstable, it gets misfold, and it forms amyloid fibrils. It cannot pass to next generation. This thyroid amyloid deposits in the wrist. It causes carpal tunnel syndrome. If it deposits in spinal cord, it causes spinal stenosis, and if it deposits in heart, it causes heart failure. So there are three types of diseases called can occur because of misfolding. So loss of function, gain of toxic function diseases, misfolding diseases. So if there is a loss of function, the disease can occur cancer, cystic fibrosis, carbonic anhydrase 2 deficiency syndrome. The protein precursors for cancer is p53 tumor suppressor. For cystic fibrosis is CFTR. Gain of toxic in Alzheimer disease, there is a amyloid beta protein. For type 2 diabetes, it's IAPP. Parkinson's disease is the alpha synuclein. And in infectious misfolding, it's CJD. It is due to prion protein. Cystic fibrosis. Uh, in, um, in our cell, there is a membrane. Cell membrane. If there is a channel called CFTR channel, cystic fibrosis transmembrane conductance regulator. This channel helps to transfer of chloride ions. But if there is a mutation in this channel, so what kind of mutation there is? If there is a deletion of phenylalanine residue at 508 position, and it causes and this causes improper protein folding. Most of this protein is then degraded, and its normal function is lost, and therefore the chloride ions cannot pass from this membrane, and it causes severe damage to respiratory and digestive system. You can see in the image, in normal CFTR channel what happens. There is a chloride ion exchange, but in if there is a defect in CFTR channel, there is no exchange of chloride ions. Alzheimer disease. If there is a misfolding of amyloid beta protein and the tau protein, then it causes Alzheimer disease. So amyloid beta protein deposits extracellular. You can see in the second image. Um, on the extracellular of nerves, there is a deposition of amyloid beta, and in the intracellular, there is a deposition of tau protein. Amyloid beta results in the formation amyloid plaques, and the tau protein results in the formation of neurofibrillary tangles. So, in the first image, you can see there are how the tangles form. There is aggregation of tangles, aggregation of tau protein, and it forms tangles. So what tau protein does, what tau protein does in the normal body, uh, it stabilizes the microtubules, and in Alzheimer's it gets misfold and it loses its function. It causes and this Alzheimer disease causes progressive causes memory impairment, inability to think by themselves, and memory loss and type 2 diabetes. The precursor protein is islet amyloid polypeptide IAPP. Am I audible? Audible?
Yes.
Yes, yes.
Okay. Thank you. So, type 2 diabetes, the precursor protein is islet amyloid polypeptide, IAPP or amylin. It is co-secreted with insulin. And the toxic aggregation of IAPP, there is a dysfunction of beta cell, and then there is a weight gain or weight loss, excessive hunger, poor wound healing. You can see in the image there is a region which is responsible for amyloid formation. That is, this region there is a mutation and it causes the misfolding of this protein. Then Huntington disease. There is in our body there are 5 to 35 repeats of trinucleotide CAG, and this sequence produces Huntington protein. And if there are more than 35 repeats of CAG trinucleotide, then it causes Huntington disease. Then faulty Huntington protein, it interferes with nerve function and it damages the nerve cells. Symptoms are slow abnormal eye movements, involuntary jerky, muscle problems. There is a enlargement of ventricles in the brain in Huntington disease. And Parkinson's disease, it is mutated form of alpha-synuclein causes Parkinson's disease. The main role of alpha-synuclein is not yet discovered, but the ability of alpha-synuclein to regulate the flow synaptic vesicle, and hence it compromising the signaling between the neurons, and it causes the loss of dopamine in brain. And symptoms are resting tremor, muscle rigidity, postural instability. Prion protein, these are small infectious particle. Uh, it causes protein called as prion protein. They are mainly found in the central nervous system. And that that transmissible, and this is caused by the prion protein are called as transmissible spongiform encephalopathies. Uh, they cause the mainly brain related or spinal cord related diseases. Uh, and the prion protein has the 40% extra beta sheets and it makes it protease resistant. The disease occur in both human and animals. This, the prion disease is Creutzfeldt-Jakob disease, CJD, neurodegenerative disease. It destroys brain cells and causes tiny holes in brain. That is healthy. You can see in image the healthy brain and the CJD brain. Uh, in this, brain becomes like sponge. The symptoms are difficulty in body movements, memory loss, etc. Thank you.
Okay. Presentation. I mean, there were many examples what she has quoted, and some different type of the examples. So what all of you can do, that you can make a table, that the name of the disease, which protein is the misfolded or the defective protein point, so that you can put on. And then next column could be that what kind of defect is there, means whether the hydrogen bond formation is not proper, or disulfide bond, or some other, whatever the kind of defect which is there into the misfolding, that you can put down. And at the end you can put that what are the symptoms and the other things which are required to know about a particular disease. Fine. So that way you can prepare a table, because if your exam is going to be MCQ type, it could be asked about a particular protein involved in a specific disease. So something like that. So you should be prepared by both the angles, the subjective as well as objective. But the two most common examples which are already dealt with when you're talking about protein misfolding, the one which I already told you about, the sickle cell anemia. So that is one of the most favorable, not favorable, but most favorite question which is being used by teachers or the faculties. So you add that particular example also. So there it is very easy to understand that even a small change into a small nucleotide or one single nucleotide is going to affect the amino acid residue, which is going to affect the overall protein structure and its folding. Right. So that example you can add upon. And the another favorite question example is about the cystic fibrosis, which he has already told about, uh, the proteins which are involved into that particular disease. Right. So that way you can prepare a good note onto this with respect to the protein folding and the what you can say, the misfolding, not folding, the misfolding, and what kind of diseases it may lead to. Right. So as such, there are no questions which could be asked from my side, but if anyone is having any type of a question or a doubt, you can just put it on. Yeah. Any questions? Meanwhile, write your roll numbers in the chat box. Nupur, write the numbers and send me across.
Okay, sir.
Yeah, any questions?